is a seminar, VTLSS seminar. And today's speaker is Dr. Winnie Curtis from Penn State. Dr. Curtis actually have more diverse training background, and he's doing work interdisciplinary research actually. So he actually started bachelor degree, started with biochemistry, but end the degree, get a degree in chemical engineering. And he moved to Purdue University and started working again very interdisciplinary research. And he has 3 advisors, one advisor in horticulture and one advisor in biochemistry. And the major advisor
pharmacy. It's a pharmacy.
Advisor is a chemical engineer. So even most of his research done in Department of Horticulture, right?
Yep.
But he got his degree in chemical engineer. So very interesting thing is Dr. Curtis actually tried to finish his dissertation, right?
Yeah.
And at the same time already started set up his own lab in Penn State and advising graduate students actually. It's very unusual. And at Penn State actually, Dr. Curtis also worked very diverse thing actually, including 50 organisms actually. Because he's working chemical engineer, he worked on plant engineer, but very different from most of our plant biologists actually. He probably really applies the engineer concept applied to engineer plants. So I bet Dr. Curtis will give a lot of examples how he do genetic engineering plants. It's a little bit different from our plant biologists. And Dr. Curtis also get a joint appointment in Department of Biology and also chemical engineering. Welcome.
Thanks.
Thank you for giving us.
Thanks, guys. Yeah. Sure. So I'm gonna, let's see, I'm mic'd up here and we're set up here and we're good to go. Okay, so this has been great, actually. I feel like I forgot what it was like to do an in-person seminar. The last one I did was, I think, 4 years ago at RPI. And then I realized that since then I've spent about $5 million worth of research and I hadn't updated anything at all. And the other thing is that I always, just the meetings with people, I kept changing my slides. Even last night at 3 a.m. I was like, oh, geez, I really need to add that one. This actually gives you an idea of the diversity. This is a little patch of my yard. I hate grass. And so I don't know how many plants you can sort of identify in that area, but the basic idea, or you're going to see this is kind of similar. I throw this one up here because I think I characterize seminars by 2 different methods. There's sort of the seminar that I give that's sort of very satisfying and focused. And the problem is that if you don't like Virginia ham, you won't enjoy this dinner, right? And so instead of that, I have the alternative. Oh no, we are recording. Yes, we are. Good. I just wanted to make sure. And it's this for those of you. So this is going to be a buffet today. It's not going to be a satisfying dinner. There might be a little bit of things that you like or don't like, but as you make your way through there, we would call it in Pennsylvania a smorgasbord, and that's what the seminar is like. I made this slide to try to give a little bit of context, trying to even wrap my head around what I have worked on historically. I am a notorious cheapskate in so many ways, but if you want to make a cheap platform, you really want free reducing power. And free reducing power is basically something where we've done a lot of work. I'll be talking about crop protection and so forth there. I will be focusing on a watershed protection for Chesapeake Bay for actually producing duckweed to feed back to animals and so forth. But we've done a lot of work historically on algae, and including both the biodiesel and really biohydrocarbons, a unique bacteria algae that we talked about here. And this also led to some work in electrofuels, where we were actually working with chemolithotrophs. So we actually use this instead of letting the algae split the water and get the reducing power, to actually use low voltage solar panels to produce hydrogen and oxygen. And then from that, combine that with CO2 and basically run photosynthesis, but run it based on hydrogen power. And so that's the common theme that I'll be going through here. The outline, oops, that's wrong computer, okay, is here. Oh, and there's a theme here of what has driven my research over the years. One is bioreactor design, having actual bioreactors that enable me to do experiments that I couldn't do otherwise. And in the process of doing it, it's also molecular biology. So you'll see those tools being the major drivers of what we've worked on. And I've had the pleasure of working with food, everything from Gates. We worked with Asian yam, Diascorea, to work with Weyerhaeuser on tree propagation. But we've also had projects on vaccinating cows from transgenic plant material as well. So I have a few comments there. I also wanted people to be aware then that I have this first slide. I call it my research collage. I've never made one of these before. It's a lot of things that I've worked on that may be of interest to you. And I have actual separate slides on essentially everything on the collage, but I'm not really going to talk about them except on one slide. And that's everything from assisting companies on killing bed bugs with entomopathogenic fungi, things like that. My first 10 years of my academic career, I was also 40% of my time as an assistant professor, which was probably about 40 hours a week, was running a bioprocess facility. So I helped companies do scale up of technologies and everything from CHO, baculovirus, SF9. I did all that kind of work as well. But we'll also have some other things there in the collage. The other is a little bit more of a philosophical, how one can take a diverse research approach to things and then morph that into the next project. Okay, and so there's going to be two of those. One's going to be in biofuels and the other is going to be in plant propagation. And it's just literally one slide each of the transition of those projects. And then I'm going to take the deeper dive into the two more larger programs. And the one is a crop gene therapy program that we ran through DARPA as the director of that. And then also a Chesapeake Watershed Protection project that's just really getting started here as we go. So the first slide I'm going to throw up here is the collaboration collage. I sort of became known on campus as if you had something and you didn't quite know how to do it, I would help because I've been helping with companies for so many years. This is a great example here. This was actually, there was a leakage in the nuclear waste facilities at the Hanford nuclear site. And then we needed to assess the growth, yield, and maintenance coefficients of geobacters so we could feed acetate into the ground to reduce the metals, get them out of solution and prevent watershed. And this was probably the most complicated thing I ever did, was handling solids in a continuous flow reactor system to assess that kind of thing. We worked on actual bioplastics. Actually, the squid ring tooth is very similar in some ways to spider silk. Only the proteins are small molecular weight, so you can actually do thermoplastics and you can do injection molding and so forth. We worked on some work here on vaccinating cows, although Allison is actually at the Military University of Maryland, so my guess is cows were not really what they were worried about, but it was nonetheless using mucosal vaccination associated with that. We did scale up in large-scale reactors enough to feed cows material for vaccination. There's some other things that are down here too. We've actually done some silencing to manipulate the pathways for CBD and THC. In collaboration with Cyram is at the Penn State Harrisburg, and he's got a whole breeding program there on hemp and some things on parasitic nematodes and things as well, which is really fun projects. You can imagine is there any entomologists in the room here? If you're not, they're doing solid state fermentation where you would go to a slaughterhouse and get all the entrails and grind them up and put them in sponges to make. And then they would have an entire barn full of sponges for artificial insects to grow insect parasitic nematodes. We transitioned that to a liquid-based fermentation, 1,000-liter scale. So they're now the world's largest, one of the largest producers of parasitic nematodes for organic gardening and for application on your lawn. So you can get rid of your grubs without having to throw a lot of chemicals on it. So that's the part one. The research collage, just background of a little bit of diversity of things that I've done over the years, which quite frankly is it's a lot of fun. The next one here is this progression. There's two progressions. The first one is in algae biofuels. So we had worked in algae biofuels, which led ultimately to a membrane protein expression, which I described very interesting because I was actually avoiding some problems in doing consulting and reactor design, which led me to work with anaerobic photosynthesis, okay, which I didn't even know existed at the time, but I'll get into that. And then ultimately to do an ARPA-E program in electrofuels as well. And they all fit very logically together, although superficially they may seem completely different projects. The algae biofuels work, like a lot of this, was based on the ability to design reactor systems for very specific reasons. These reactors over here were used to do stoichiometric assessments of the utilization of nitrogen. And so when you take a look and you write stoichiometry for biological systems, people ignore the protons and so forth. They just buffer the heck out of the media. And the reality is that algae will actually eat themselves to death on ammonia if provided ammonium nitrate. They'll just drop the pH until they die. So that's my sort of, I call it my broccoli versus Krispy Kreme donuts, okay? And so you have to play parent for that and actually do process control based on nutrient feeding. The others that are on here that I'll mention real quickly, I have a slide of each. It's a very similar looking reactor, but this is actually an anaerobic bag. So this is a bag that actually is filled with nitrogen, and they're using photosystem 1. So based on photosystem 1, it's a very efficient system for doing functional membrane protein expression. And so that's some work that we had done there. And we had adopted that from Argonne National Labs, where they were doing, this is their culture productivities here. And this was our first run using fed batch succinic acid based on pH control, and then use that for membrane protein. And then finally, this is another exciting one here. Electrofuels is actually a project where we were actually fermenting explosive gas mixtures. And it turns out that Rhodobacter is capable of that. And so we were actually using the same organism, chemolytic trophic drove, for fuel production. If you take a look now, I have basically a slide for each of those 3 areas. The first one here then is this is an ultra high density algae production system. These are actually, when you see most people working with photoreactors, they're usually running about 2 grams dry weight per liter. I'm not sure if I have a pointer or a weapon here for that, but if you can find one, I would use it. And we're operating under light limited conditions. And I think the main point of this, which is I wrote a paper, I tried to get it in Nature Biotech, and they considered it for a while, I haven't gotten back to it. I call it slow and steady wins the race. Okay, Botrococcus grows very slowly, but it turns out that its electron use efficiency is about double that of chlorella because it dumps it into isoprene hydrocarbons. And so under these conditions, we actually run under light limited conditions. You don't have to worry about the growth of your organism anymore, because if you don't operate your photoreactor under light limited conditions, then you're obviously wasting electrons. And so under those conditions, then you don't have that. The university didn't want to file a patent on this thing, so I decided to educate myself. And I went ahead and I wrote a patent. And actually then eventually one of my buddies from undergraduate, it turned out to be a patent lawyer. So when I got to the final stages, he helped me iron out the loose ends. So that was actually awarded. So I have a patent on that. And the real interesting characteristic of it, this is the only reactor that I've found that operates at these productivities under ambient CO2. You don't need any CO2 supplementation to this system because it will actually, and if you think about it for long-term sustainability, if your system doesn't actually use ambient CO2 and you need a coal-powered fire plant to get your CO2 to run your algae reactor, it's not really sustainable in the long haul anyway. Okay, so let me find my mouse again. The second part of this one here is, okay, so this is the slow and steady part. So this is actually the oil productivity. So essentially you can think of this as energy, only this is more energy dense than this is, even on a weight basis. And so at these productivities, the Botrococcus brani, which makes C34 branched hydrocarbons, it actually accumulates the energy much more efficiently relative to chlorella at the same density of cells. And so that is the rationale that I was trying to get across. We did an SBIR actually at our family farm down in central PA, probably the worst place to do this in October. Okay, but just when the SBIR money was available, so we built an outdoor prototype system on it. And what it came down to, if you wanted to actually predict growth, it was nothing about growth kinetics or anything. It was just about finding out when the sun was on the panel. And you could actually then predict the productivity of your system based on just solar azimuth and how tall our mountain was behind the sun, whoops, and so forth. So that's the algae side of things. The interesting thing was that when we were doing this work, I was doing consulting for a company that was doing it was green fuel technology. They'd acquired about $100 million to do scale up of algae biofuels. And they didn't want me to do reactor design for them because they thought that would be a conflict of interest. So what I did is I thought, great system, I'd like to develop it more. So I did it for Rhodobacter because Rhodobacter only has photosystem 1. And I read my contract very carefully. Okay, it talked about photosynthesis and algae and so forth. So we could do the same type of technology development, but do it for a anaerobic photoheterotroph. And so the neat thing about this organism is that when it goes anaerobic, it lives at the interface of where organic acids come out of the mud and the infrared light can make it down through the algae and so forth. It is capable, it can actually produce 5+% of its dry weight will be this LH2 light harvesting pigment. And it fills itself with intracellular membranes. Argonne National Labs had been trying to develop this, but they had never tried it with a photosynthetic system. And so what we did then with this is we went after aquaporin as a basis for a membrane, functional membrane expression system, and then expressed that in this particular system. We then wanted to do the first actual measurements of the transport rates of water through the aquaporin transporter because it had not yet been done. And so to do that, we used stop flow. We actually have reconstituted the aquaporin into vesicles, ran it through a stop flow, and you kind of think of this as these running at 2 different rates, and then you have a constant residence time. And at that time, you can actually measure the shrinkage. The shrinkage will actually tell you how much water was taken up. And then we actually use fluorescence correlation spectroscopy then to go ahead and do measurements then of, the way to think of this is a molecule enters this 6 femtoliter volume. And if it manages to get across there before it gets the fluorescence resonance, you can measure essentially diffusion coefficients and number of molecules. That gave us basically, we knew the amount of water, we knew the amount of aquaporin, they're tetramers actually that make that. And from that, we could actually calculate it. And I love to actually express things in the end in terms of, how many water molecules do you think a second can go through aquaporin, noting that aquaporin is able to actually not dissipate proton motive force. So the water has to do a dance through positively charged amino acids. And that answer is 10 to the 10th molecules per second. And so I like to express this to students when can you start to think about how fast biology has to work in order for you to not explode when you drink salt water. So that's the magic of aquaporin in this particular system. And a little lesson to you there is that you can't have all the toys, okay? Because these toys, I didn't have a stop flow and I didn't have a fluorescence correlation spectroscopy, but we had the ideas and we're able to use those tools to put together the actual measurements themselves. Rhodobacter is relatively unique then, is that it is also able to do chemolytic trophic growth. It's considered to be one of the most diverse organisms on the planet. It can be heterotrophic, anaerobic, photosynthetic, and also chemolithotrophic. Okay, so it's the junk pile of bacteria. Basic idea is photovoltaics can actually split water at very low voltage. You just need an overpotential of a volt or so. And then you get hydrogen and oxygen, combine that with CO2, and you run a reactor, which is very unusual because you actually feed it stoichiometrically. It eats all the gas. Unlike a thing where you blow a bunch of bubbles through there and try to get a little oxygen to transfer, this is actually stoichiometric utilization of the actual gas itself. We worked with Joe Chappelle. He's been a collaborator with me since I started in 1988. He and I started some things on sesquiterpene cyclases and fungal elicitation, but he actually had the pathway for this since we actually genetically engineered the hydrocarbon biosynthetic pathway into this chemolithotroph, based on the classic MVA, MP, MEP pathways and so forth, and then used that to literally ferment an explosive gas mixture into hydrocarbons. And so this is actually was in biotech bioeng, and we actually then looked at things like the production of the hydrocarbons, carbon balances, energy, and so forth, associated with the fermentation, if you will, of this particular system. So that ends the first one, okay, shall we say, the run through a development. This is propagation bioreactors. Actually, the very first grant I got as a faculty member was a whopping $50,000 to develop somatic embryogenesis, where I just made the observation that we tend to take plant tissue, put it in, and then solidify agar, and then take tweezers and move it. And I was like, that's like the dumbest process I've ever seen, right? I mean, why wouldn't you run the liquid past the solid as opposed? And so in that was the context of some of the original reactor work that we did, was literally inclined planes, where we would run programmed hormone development of auxins and cytokinin ratios to actually manipulate the formation of embryos. I never published that, but some people saw it at a meeting and they invited me to some meetings, and then we went on with it later. But I did get a big grant with NSF to look at transcription factor manipulation. And so the basic idea is reactors, you can take, hormones and physical chemical conditions, but this is genetic manipulation you can add to the reactor system. And that was the basis, which ultimately led to projects that we did with the Gates Foundation on African food security. So that progression, there's a few others, there's the classics. We did some side projects with Nunham seeds, if you've ever wondered about where seedless watermelons come from. Okay, one way is actually through clonal propagation. We did work with Weyerhaeuser on synthetic seed production as well. And the work on transcription factor manipulation was done with cacao. And again, one of the things that there's not too many young faculty here, they should be here. You guys should slap them. The point is that I always thought you would work with the hardest possible system you could come up with. So they discover baby boom in Arabidopsis. I'm like, yeah, let's go to a woody plant, okay? And it worked. I was fortunate in that context. But nonetheless, and then the work with the African food security, it was basically applying the principles we learned here to doing propagation of yam. And so I have, again, 3 or so slides here that'll get us through this. The driving force for Weyerhaeuser's thing was take a tallest, straightest tree that you can find in the woods, you plant it, and plants maximize variability. You get back the same population back again. And so their idea, of course, is find the tallest, straightest tree, clone it, which happened to come from embryonic things, stick it in cryopreservation for 10 years, do field trials, find out which one is the tall tree, and then you would have the superior productivity trees and so forth. So cloning was really the basis of this. One of my favorite pictures in the history of my lab is this one right here. Okay, this is actually a filter paper. If you look at it close here, and those are baby pine trees. Okay, and isn't that cool? It really is. Okay, and these are embryonic suspensor masses. This was developed, the technology we done Darzan at Davis, and then I worked with them with a goal of coming up with a reactor system where I could go from a cryovial to 10 liters and about 10,000 trees in a rolling carboy. And that system we had to overcome mass transfer limitations here and also use carbon dioxide to reduce stress, not to do photosynthesis, but also then do oxygen supplementation to overcome mass transfer, because as these embryos start to develop, it turns out if you do the calculations that the meristem that's dividing here, the rate of respiration of a plant cell is about taking a jog for a human being. That's the tissue respiration rate. So clearly their oxygen transport limited there, and we could actually show we could double the rate of growth by doubling the partial pressure of oxygen in the environment. And you can imagine for rolling a carboy for a couple of months sterilely, where you're controlling the gas phase environment and so forth. That's one of the toys that we were working with there. The transcription factor manipulation, the next phase of this was the basic idea. This crowd, normally I have a bunch of engineers, and when I start to use the word clone, they kind of, what? Okay, it's like, yeah, just take a couple of your cells for your hands, it's somatic cells, turn them into another one of you, okay? why we would ever want to do that with faculty, I have no idea, no. Okay, so we went ahead, found the homologs for the transcription factors here, made the cDNAs and so forth. You can see Arabidopsis is over here, and the Theobroma cacao chocolate tree is there. From that, because we wanted to use homologous transcription factors associated with this, this was about, goodness, it was in 15, so this was probably about 2008, 2009 when we were doing this. It was very long before what's going on now. They weren't even called morphogenic regulators. And in making transgenics here, you can see in the presence of doing transformation, you could get a lot more embryos, and they looked a lot happier as embryos as well. But the fundamental problem that you encountered here is that it's kind of like the it wasn't controlled, right? And so we ended up with them wanting it, I guess, like faculty or administrators, I'm not sure which, want to be like infants the rest of their life. Okay, and we needed a way to turn off embryogenesis in that. And so that led us to looking at alternative ways of doing this with dexamethasone. So Elliot Merkowitz had developed the dexamethasone-based system for induction. So we got that from him at Caltech. And use the LEC2 promoter. I don't know if did you find a pointer, or we just go with the finger here as we go? There's no batteries. Okay. I should have brought my own toy, but that's okay. The LEC2, it's a transcription factor, so it of course binds to DNA to manipulate it. However, the glucocorticoid receptor binds with the heat shock protein and that keeps it out of the nucleus. So if a transcription factor, even if it's constitutively expressed, is not in the nucleus, can't do anything. When you add the dexamethasone, it displaces that, lets it get into the cell, and that gave the basically an inducible system for doing that. So we screened through about 2,000 embryos because making a transgenic of cacao is like the most miserable thing that we did. But we managed to get one transgenic, and that one transgenic then gave us ultimately a, oh, okay, so let's see if we, I don't want to blind anybody, there we go, okay. And so, you get the binding and so forth. This is actually a dose response to dexamethasone. And this is the number of embryos and time. And, just dose response. So clearly it's a transgenic that's responding to the amount of dexamethasone that's in there. And then you can take a look then. Our real goal, remember, as an engineer, is I wanted to take a leaf, stick it in a blender, throw it in my reactor systems and make thousands of trees. That's what I wanted to do. And so we did steal some leaves off of the trees, throw them into their dexamethasone, and demonstrate that we could actually go directly now from a somatic tissue like this. Instead of the classic way of doing cacao is you had to dissect, like before the flowers opened the stamens and put it onto auxins and cytokinins and wait 6 months and all that other stuff. These guys popped up in about 10 days. Okay, so that was the basic idea of using transcription factor-based systems for that, for enhanced embryogenesis. Argument, of course, is the real motivation for most people in this technology is transformation, right? But our goal here was really not transformation. It was actually production. I threw this slide in late last night. It's something that I've thought was really, this was a cotton. Cotton, they had actually discovered a phytocyanin like a arabinogalactan. I called it magic dust, which you actually add on to plant tissue culture, and you can actually dramatically improve somatic embryogenesis. In fact, we took the cotton and stuck it onto cacao and saw behavior there as well. I've only seen one paper since that. It's been 10 years ago now, and they actually showed they could take it and they could put the cotton onto, I think it was tobacco or something. So it seems to be very broad. I think it's very interesting from a scientific standpoint here that I think because this has no hypothesis as to what it is, nobody's followed it up. For me, it's like the perfect thing. I mean, I can, oh, by the way, you can express this PLA in E. coli. And so it's nothing to do with the actual arabinogalactans that are on there and so forth. And so we actually did some work with that, and I've been trying to find the time to publish it, but haven't for a while. Again, I'm a technology user. You guys are the ones that make all the discoveries, and then I just steal them and use them for things. That's my job as an engineer, right? And so now Gates Foundation here, we were trying to propagate yam. But the goal here was inexpensive. Okay, the technology of doing gas phase manipulation, which was our key, because if you think about it, why do we even have to put sugar in plant tissue culture media, right? And the reason is because the lighting levels that we use aren't enough to drive photosynthesis without heating up the vessel too much by the greenhouse effect to kill them. And so we literally have created a system that's contaminated by sugar, okay, because we're trying to not overcook our tissue culture at the same time using quote unquote cool white fluorescents and LEDs. But if you're smart about it and you can elevate the carbon dioxide concentration in the headspace, you can actually take the sugar out. Okay. However, if you try to do that for the long periods of time where you're mixing gases together and so forth, it turns out the expense of that is more expensive than the light bulbs and the sugar in the first place. And so we're actually working on how one would use low-cost automation and that for these types of production systems to just simply raise and lower this thing with a stepper motor. And then these are automated systems for production. And we just finally got around to about 6 years worth of work to publish this. And this was the, we were demonstrating, and this is the Asian yam, the project for the Gates Foundation. Well, you'll notice this is no sugar, okay? And so our goal from here was to take this into, during COVID, unfortunately, we were going to do an actual demonstration and look at, and testing in Africa. And that all got a little bit sidetracked by COVID. But nonetheless, you can kind of imagine that if you then get the lighting right, you can do this in a greenhouse setting. And so the idea is to get to very low cost propagation-based systems where you still have some control of the environment, gas phase, and so forth. And you can probably slip in a few transcription factors or magic dust if you need to as well, where the basic idea is productivity-based system for propagating. It turns out that because if you see, let's see if I can do the back there. Wrong toy. Okay. Oh, well. I guess we won't do that. Oh, yam is kind of like potato, and it's passed from chop a few things off and then give them to your neighbor kind of thing. And so it's got lots of viruses and disease and so forth associated with it. And so this was a supply chain thing, just like our French fries for McDonald's go through virus index potatoes that are then grown in virus index fields, and that they don't have anything like that in Africa. And so the idea was to come up with supply chain associated with plant production in Africa. The DARPA project here is one that was like the biggest project, and I don't know if you want to call it blessing or nightmare that I went through, was putting together a project on executing gene therapy for plants with a unique aspect. For human gene therapy, we use attenuated real viruses, and then we stick you with a virus. And that's where the danger comes in, and occasionally people die and people get excited about that. Plants, we can genetically engineer. So if I could genetically engineer and put virus functions in you, I could then make my virus debilitated so it wouldn't actually have virus functions anymore. And so that was the basic concept of this particular project, was to basically then take the plants, use insects as a vector to that to deliver therapy to plants. We actually focused on the artificial microRNAs that we were actually delivering to attenuate drought stress. So we could control the plant's response essentially to various stresses. And here's the crew that was willing to put up with me running this project. One major cog was missing here. The major cog was that of the administration, getting the reporting of finances correct and so forth like that. So in the final third phase of this, we were literally 80% done this project, and DARPA had to pull the plug on it because they couldn't get the finances right. And so you can kind of imagine what I'm going to show you now is the aftermath of having to terminate 12 postdocs and then try to finish up the rest of the project as best we could during COVID over the last 3 years. So I put that in context because there's still a lot of really cool stuff that we got from this. This is really a neat project. If you want to think about virus complementation, the concept's pretty simple. You take your virus, you put the function in the plant. That plant then turns on that functionality only if it needs therapy. And therefore, if you have a drought-inducible promoter, it'll come on. And then you place the protective genes and so forth in here. And then you end up with a virus then as a deconstructed virus that needs that plant in order to function. And it only functions if the plant is actually under disease and stress. We got wonderfully written up in Science as potentially developing bioweapons. And then I had to deal with that for a few months of damage control. But the basic idea is based on this idea of the insects. And we were working with not only trying to make conditional control here for biocontrol, but also insect conditional lethals, as well as containment of the system itself. Where we started with this was actually to identify stress. So we worked with PNNL, and they did, we have 20 times this data because this was the very first thing we did was a simple proteomic analysis in phloem exudates. We've actually now done it for entire drought stems versus veins versus leaves and all that stuff. We haven't published any of it. I think I have someone in Israel that's going to help us do the final proteomic analysis soon to send that out. But the one thing that we found here was one of these genes here had 130-fold. TAS14 is actually the driver of hydrophobin. And that hydrophobin gene then became the basis of us because we needed to get the ball rolling and making all the transgenics. And the transgenics that we were working with here. So we actually then took 3 different truncations of the promoter because we didn't know exactly what we were going to be working with. And then we were going to derive different forms of the virus replicase functions, the coprotein, things like that, off of this promoter. And then ultimately come up with showing our ability to turn that functionality on during the actual stress itself. Okay, so that's the idea behind the actual project itself. I fully admit I now really appreciate real breeders because there's my risk. I got 100 different constructs we made the transgenics of, and we're doing digital PCR, where if you're not familiar with that, you do an aerosol. And then that aerosol compares to a housekeeping gene, and then you get the ratio, about 1,000 reads per aerosol drop, and that'll give you the digital PCR will give you the ratio of your gene insertions. And then we do the segregations of tomatoes, which are very fun. I own all the windowsills of the entire south side of the building because I don't even have a greenhouse. And so I do have a biocontainment greenhouse in Harrisburg, but it's an hour and a half away. And so a lot of my tomatoes, I just grow them out on the windowsills, which is a concealed building, so we're good. And we have then here, this is the the typical run through the process. We are literally right now at the final stage of homozygous for about 20 different constructs and half a dozen transgenics in each ones for the tomatoes that we managed to wiggle our way through at the project. Now, okay, so this one here is plants that have the virus replicase. I say replicase, and it's not really true, right? It's a replication-associated protein, but pardon me for the virologists in the room. Okay, but it's that replication function is turned on. So now what I need to prove the utility of this is actually a virus that doesn't have the rep. We have a lot of different configurations, but the one that's the most, shall we say, easiest to think about is we just have I'll explain here in a second. If you make a 1.5 mer of the virus, it's genome plus another one. It's a single-stranded DNA virus that kind of wraps around itself and does rolling circle amplification. And so we actually can take that and just basically knock out the ATG. And so what you have is essentially the exact same virus. We're not messing with the genome that much, but it still doesn't have the actual start codon to make the actual replicate the rep. And so the rep then, we actually then showed the ability to do complementation. And so this is actually a virus then that has the replacement here of the coat protein is GFP, and you can actually see then complementation does occur. To understand, how this technology is then based on a lot of different elements, if you can kind of imagine. How would I launch a lot of viruses that have been genetically engineered in a plant? Fortunately, with the begomaviruses, what you can do is that this is a pepper golden mosaic virus. This is its genome here. Okay, and then here's the coat protein replaced by luciferase, and then here's another half of the genome, if you want to think of it that way. And so what happens is we place this on a binary vector, and then you can agro-infect that into a plant, and then what it'll do is it'll launch, and it'll actually, they're called infective clones. And the infective clones then will give rise. This is a bipartite virus, so it has an A and a B, so we have a 1.5 mer of each one. We do the agro-infection into that. It gets up and rolling in about a week, and you can have a full-blown viral infection, which, if there's anything scary about the DARPA project, was revealing what we do as scientists. It was not inherently. It's like, oh yeah, you can use agro to launch TYLCV and other things like that. Well, yeah, that does sound a little bit like we should be concerned. The 1.5 mer, the way it's designed, is had a nice convenient spot to replace the genes in there. And then we got really caught up in working with everything, the M-neon green, M-cherry, artificial microRNAs, nanoluciferase, also mushroom luciferase. And we actually made intron-containing ones of all of these. And if anyone wants any of these toys, they're going to be available, and they're going to be an ad gene. And we're doing full sequencing with Plasmidsaurus of everything, so the toys should be available. Here's just one showing the pepper golden mosaic virus that has a GFP replacement, if you will, for the coat protein launched into a plant. The next thing we need to solve is how we're going to get the virus into the insect. And so we started here with actually coming up with membranes, and we wanted to do this aseptically for reasons I'll explain in a minute. This right here is actually a Texas red. And then there's a membranes. We tried different types of membranes and so forth. And this is a confocal image then showing that it's actually feeding on through the membrane, which we had an asymmetric membrane because it kind of looks like a leaf, and they literally feed up through the Teflon membrane for that. Anyone that works with insects, a lot of times they use these like parafilm sachets, okay, which is great if you're going to work with parafilm, but if you're going to autoclave it and try to scale it up, parafilm isn't going to work. And so we actually then use this technology then to launch the virus, do a real crude sep, put it in 150 grams per liter sucrose, a little bit of yellow dye to make it look like a solanaceous flower, and off they go, and they feed on it, and they actually take up the virus. Now, one small thing was missing from this, and that is that we needed to come up with a sterile colony of insects, okay? Being naive is a real advantage as an engineer. I'm like, oh, sure, we can do that. Then 2 years later, we figured out how to do actinic whiteflies. How do we do it? It turned out that this was a race between all of the, it's a phloem-feeding insect. Every time it lays an egg or sucks on a plant, it introduces pathogens and so forth. So we ended up having to grow phototrophic plants, get them onto there, get them to lay an egg and get through their life cycle in less than 21 days when the fungus is trying to make its way down through into that after surface sterilizing it. And we finally came up with a way of, with a simple GA7 now, we can produce 300 whiteflies in about 5 weeks from half a dozen. And so we can literally do the entire maintenance of sterile whiteflies in a tissue culture-based environment, which used to, when we budgeted this for the DARPA project, we were budgeting about $100,000 a year for the technicians, the lab space, the greenhouse, and everything else. I now have an undergrad that I pay about $5 hours a week that maintains our sterile insect colony. So I'm a cheapskate at heart, so that helps in my efforts here. And so this is actually some looking at host range. And so here we were taking a look at how many I could catch. Okay, on the top of the actual GA7, just pop another one on top of there. And I did that, every day. And you can kind of see there's cabbage is great, it turns out here, and sweet potato. Ironically, tomato was not so good. Tomato is actually hidden down here. And the reason for that, actually, it turns out in tissue culture, tomato doesn't tend to expand its leaves. And as a result, the trichomes are really dense. And as a result, they can't seem to feed. But it turns out we found out a way around that by actually growing them phototrophically and leave the sugar out. They actually will expand out and you can actually proliferate in that. And then there's some other tricks we went through for looking at that. We actually then made a kinetic model assay for the proliferation of flies. And so this is a little tiny one of the many, many equations here. But what we could do is we could actually take a system and then we could, after a certain period of time, we could back calculate how many of the flies were viable. So we made a fecundity assay out of the whitefly colony maintenance that we were working with here. And that gave us a way to either predict forward how many flies should be there or predict backwards how many were viable at the time of introduction of the flies into the system itself. And that's actually published by she's the one that inherited all the unfinished work. And so Natalie is a fantastic PhD in my lab. We got a little carried away with trying to look at host range of this. These are all tissue cultured plants. We're kind of like, hey, what can we grow these on? And it's pretty interesting how it works out. This one was really interesting because this is related to the food security in Africa. Two different, I guess, call them genotypes or species because Dioscorea is a little weird. One had very good proliferation and another one did not have proliferation. And so there's, inherently is like, hey, I wonder what this is. Is this physical? We were actually looking at trying to use an ATM, atomic force microscope, to see, is it a matter of they can't feed physically? Is it a physical problem? Is it a chemical problem? That kind of thing. And we also did some preference studies, which I thought were kind of neat, where you could put two different tissue cultures in the same one and then find out where the flies went, which ones they liked better than the other. And ultimately then, so this is just, so now we have a system to introduce a virus, and we wanted to actually have a plant that we could use to validate some modeling. I'm not the modeler, there's another guy in the lab, he likes to have 20, 30 differential equations. And we wanted to look at virus movement in plants. And to do that, what we wanted to do is actually take, in the virus, you have this transactivating protein that comes around to the promoter, turns it on, and turns the co-protein on. And so what we do is we actually take that promoter, and it normally has a plant protein on there that actually is a repressor, and the trap actually displaces that. And so by taking the luciferase on this, then what we end up doing is making transgenic plants that would turn on, if you will, the luciferase when the transactivating factor was present. And so we want some way to actually measure, if you will. We actually have another thing I have looking at, since this is a bidirectional promoter, is putting two different reporter genes in opposite directions. And then we could actually be assessing both the forward replication type function simultaneously with the encapsulation function based on a reporter gene that was being transactivated in the actual tomato itself. Okay. So the next slide is just for fun. And so it's a little bit about the idea of insecticidal plants, and that's just just to see if you're awake. Okay. The basic idea here was to try to sterilize insects as they fed. Okay. And so what we did here is we took a chitinase and we took a synthetic vitiligellin. So I took the sequence associated with the target. If you want to think about eggs, actually need to load a lot of protein in insects and frogs and things. And so it's made in other places and then it's transported into the egg. And so that was a synthetic sequence that I made there. And then this is a protein transduction domain from Drosophila. And so the basic idea here is as they were to feed, it goes up, the protein transduction domain will have it transport out into the hemolymph from the gut. And then from that, then the vitiligellin sequence will then take the chitinase to the ovaries. And then the chitinase in the ovaries will disrupt the fecundity of the female. And then we've done a ton of this. These are still going through the segregation process and that. And then went back to, ironically, my original fungus that I was working with, opium poppy, as my PhD, and Rhizoctonia solani and fungal elicitation. Well, it turns out it's an actual chitinase assay. So you can actually put the chitin there, and then you put your extracts, and then you see an inhibition of the of the expansion of that is actually using as a bioassay associated with this. I think the real fundamental problem and difficulty we have at this is it is hard to get reasonable expression levels in tomato. So we were forced to work with a tomato-based system. It turns out that we now, from George Jander up at Cornell, we have an acyl sugar knockout that he had developed as part of this project too, that whiteflies can now feed on the Nicotiana benthamiana. And so now we have a Nicotiana benthamiana system. We can do the launch, we can do the feeding and full cycle and so forth. And so we're going to probably go back and then retransform some of this stuff into Nicotiana for proof of principle as well. This is the last slide, I believe, of the DARPA before I get to the duckweed. How am I doing on time here? Okay, oh, it's too late. Okay, if you need to leave, feel free. When I developed this system with DARPA, I told them I didn't want to build a car that I couldn't turn off. And so I wanted to develop then from this a kill switch to be able to shut down a viral infection. The basic way in which we had done this is we actually took the trap gene here. Okay, it turns out that what we did is we made a silencing RNA. That silencing RNA would target the trap gene for transactivation. And then we could recode this non-overlapping section of that so that it wouldn't shut itself down. And so the kill switch is basically a virus that parasitically uses the capability of this virus to make the kill switch to shut down the actual gene that we had itself. And so we did, the proof of principle was done actually by Gary down in Texas. This is kind of classic siRNA. And you can kind of see if you introduce the siRNA, it'll really shut things down. And then the question then is that can we actually then do that with an actual viral vector? And so the green here is the viral vector relative to the one without the kill switch present. And you can see that obviously it's starting to take off and the viral vector is not being able to amplify under those conditions. And conceptually, this actually would be an antiviral, not for a wild type virus as well as for the protective virus, if you want to think of it that way. Okay, so we're almost done. We have to do a little bit of watershed protection. And you guys are about, I don't know, about 30 miles out of the Chesapeake Bay, and we're about 20 miles in the Chesapeake Bay, as I'll show you here in a minute. But the basic concept here, Rachel's actually the PI of this, and the idea is to actually take the manure runoff, grow duckweed, feed it back to the animals. So, okay. But the idea for me was actually to improve the amino acid composition of that so you could reduce the amino acid supplementation you do for cattle for production. Here's the Shenandoah Valley. Okay, so, and interestingly enough, if I'm a chemical plant and I have a pipe, I'm highly regulated for my emissions, okay? But if I'm an agriculture, okay, I'm not, okay? And so the vast majority of what ends up in the Chesapeake Bay is actually fertilizer, manure, and so forth from that. And so the idea then here is, for me, nitrogen is an opportunity for growing something, and duckweed is an opportunity to improve amino acid composition in animal feed. And so that was sort of my look at it. Yeah, I did this on my ride in. I pulled over on Route 81. It was like, oh, just outside of Roanoke. I wasn't sure if you were in the Chesapeake Bay or not, and you're not. Okay, so it's close. Okay, it's close by. It's all the same world in the water. So, but nonetheless, you should care. Basic idea here. If you look at the back end of a cow and manure spreading and so forth. It's not too surprising that a little bit of it ends up in the Chesapeake Bay. I look at it as if we can capture that and I could do actually transient gene expression, so I don't have to make GMO-based duckweed. We had originally developed transient gene expression for high throughput screening for Monsanto based on oxytrophic strains like 20 years ago. And so the idea is, hey, let's just scale that up. So we'll have 20,000 liters of duckweed and we'll transient express seed storage proteins, improve the amino acid composition in that and feed it to cows. I'm also quite interested, it turns out, in trying to use this to actually grow azotobacter or other organisms that could be used as a soil supplement. So we have some other things that we've been chasing a little bit. Too many toys here. Wait. I want to hit this one. Yeah, there. No, that's not good. Okay. Okay. So we've got a few. We cloned a seed storage protein out of cotton. And that one, you know, we're going to increase lysine, methionine, and histidine. I mean, that's what they feed cows. And it turns out it's very tricky for a ruminant. And I'm learning an awful lot about it. It's actually a physical problem. You have to actually get it to go in and not spend too much time in the rumen. You need to get it to bypass that to get into the intestines, but we have a cow guy, so he can help us with that. And this one here we just got from Israel. So zeolin was actually a chimeric protein between phaseolin and zein. And of course, the idea is to place that on viral vectors. And we are making transgenics because we want to have the material to do some testing in their in vitro. They have some like pseudo cow fermentation methodologies that allow you to do feeding studies. But we also want to do transient expression as well. We actually also, it turns out Marquita Landry is someone I've known for a long time that's done the nanoparticle-based delivery type systems. And we're going to do a little bit there to see if we can tweak the genes in as well. This is a kind of a classic one of like duckweed. It turns out has to sit in the sun all the time. It doesn't get a whole lot of shade. And so we started to look at the autofluorescence profiles and that we knew we were going to run into some problems with reporter genes for fluorescence. And so this is actually a scan going along until it basically saturates that. And clearly, if you're looking at signal to noise ratio, Cherry actually was the one that worked the best. So that's our candidate there. And so here's where we had plant codon optimized. We got a hold of, we got this. And it turns out that before they had even published anything about this, I knew about the Russian group Planta that was doing the luciferin mushroom biosynthesis pathway about 6 years ago. And they gave us the genes to actually then try to do synthesis of luciferin inside the tissue so you don't have to do the addition. So we could actually do in vivo luciferin synthesis. The way we were doing that is actually to take this pathway and then we put self-cleaving inteins in there. So we have one cistron, and this is like a polycistron for a eukaryotically driven 35S promoter driving the biosynthetic pathway for the recycle of the luciferin. It turns out tomato is a terrible system for us to develop this. They now published in Milkosiana benthamiana, and it works really good there. So one would have to do a lot more trouble there. But this is the pathway, and the kind of neat thing about it is this is caffeic acid, which is part of cell wall synthesis. So your precursors for luciferin in mushrooms are right in plants all the time around the cell walls. And so I think in the future, you're going to see a lot more with the development of this because you can have in vivo luciferin and not have to take samples and buy very expensive luciferin, which is the limiting factor for luciferase-based assays. Wrong one again. Oh, this is an interesting finding because we had done so much on these development. We went back and we looked at all these reporter genes that we were making, and it was all over the map in terms of the interference we were getting from transient expression due to expression in agrobacteria. And there's always this kind of thing where people say, oh, I don't think it's a problem in my system. Okay. And I was like, okay, we need to understand this better. The answer, it turns out, is not introns, because we put introns in everything, and it's true. It'll prevent bacterial expression. But you can also do it with UTRs. And so if it turns out that if you take one of these and you put like a UTR in here, that is sufficient, and you get the right one. I can't even tell you at the moment what the right one is. But bacterial UTRs are only about 9 base pairs. And so when we start to do cloning as plant scientists, our multiple cloning site's bigger than a UTR of a bacterial system. And so we have no idea what we're doing in manipulating the on and off switch associated with bacterial-based expression. And so we've now created one where we can do a drop-in of some random sequences and things like that. We want to try to do a little figuring out of what really happens here to give us a toy that was actually predictable, as opposed to hoping that it's not doing bacterial expression, which, by the way, in many cases like this is actually the intergenic promoter of the virus, where we don't really understand it. And so we always just put it in frame at the ATG start site. Well, it's extremely active in agrobacterium. And so you have to worry about that as an actual system. Real quick, it turns out duckweed does not like agrobacterium. Okay. And so this is loss of chlorophyll. This is actually a tumefaciens that came from Mitchell Moore lab in Davis. And he did a screen of his collection of hundreds of agros and found one that was non-necrotic. And they published it and nobody ignored him for the next 10 years. Okay, well, I didn't ignore him because I want to play with this actual agrobacterium because our duckweed gets very unhappy when we put it in. It's not too surprising if you think about it. If I have a bunch of little babies inside and all I need to do is explode to protect them, it's a no-brainer. So the hypersensitive response is probably pretty active there. This is actually where I had contacted someone that you guys know, Bing Yu, and it was like, oh, I saw his cool system. We'd actually been working on SAC-B integration into agrobacterium with undergrads for about 10 years, and we never got it quite to work. And he scooped us. Thank you. Okay, so I don't have to mess with it anymore. Okay, but trying to do that, this is actually taking now the non-necrotic agro. So we're trying to make the stealthiest agro possible for gene delivery in agrobacterium. And so we wanted to start with a non-necrotic agro. We don't even know what it is. It's okay. Okay. It turns out that Dr. Suzuki in Hiroshima had created an eviction plasmid that'll actually really easily pop the TI plasmid out. Okay. Along with a little bit of triparental mating. And then from that, actually then move the EHA 105 back in, okay? And then from there, actually use the system that Bing Yu had developed that would insert into the Rec A site. So we get Rec A minus, which is originally done by Steve Ferrand, actually, before AGL1 came out, for those who know the agro community. And then ultimately then integrate the oxytrophisms that we have. Because we did suicide vector mutagenesis, which is a terrible process, and now you can just do homologous recombination. Now, the other thing that we wanted to do is it turns out that the transcription factor work actually integrated at this point in time, because it turns out if you take a look at what the companies did with transcription factors after some of the things we had done, is they, target that one, okay, is it turns out that when you do the transfer of the tDNA into plant cells, every now and then it reads on through the border. And so they actually put the transcription factors associated with gene expression on their vectors. And all I want to do is I want to put things on there that will suppress signaling and defense. And so we got from Faith Belanger the salicylate hydroxylase that she has for her endosymbionts. And we also have one from Pseudomonas aeruginosa and some others. And the basic idea is then to create a binary vector, which actually came from Dr. Lee down at LSU, because he developed a really cool binary vector. It's only about 3.5 kb. And its copy number is 10 times higher than the ones that are in there. And it's retained even during the transformation event where your selectable marker is gone and you're losing that. So PLSU vector, it turns out he developed it, published it, again, like everyone else, everyone ignored it. I got a hold of him when I was down at LSU and he couldn't find it. He'd gone on sabbatical and no one watched his freezers and it had died. Fortunately, one of his students took some of it to Germany. We recovered it from Germany, okay? And so now the PLSU vector, which is this really small, compact, it's like 3 kb binary vector, is going to be the basis of what we build this system into. So we'll have then now a stealthy, non-necrotic, blah, 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 agrobacterium, trying to take a leverage, if you will, an NSF project on duckweed, okay, to create a tool that would be useful for the community of plant biotech is really what our goal is there. And having talked now with Bing Yu, he's got a whole lot of other nifty little things that we could put on there. So we need to get together and make the world's best agro and quit and understand how it works. As opposed to just getting my nice, pretty green pictures and so forth, they'll be optimized for gene expression. That's the end of the talk, okay? And I know that it went through lots of bits and pieces and so forth there. And I did have one last slide here, and that's as an invitation because this is my trailhead of McAfee Knob, my way down here. And then tonight I'm gonna go up there actually, and I'm gonna sleep up there tonight. And so if anyone wants to, I actually have an extra hammock and you can join me. So, oh, pictures. There's my kids, all four of them. Okay, about circa 19. This is 2022, 2002. Okay, so that's 20 years ago, and that's my kids. Okay, if I found a picture of them, and then there's what I looked like when I was a kid. And my wife has put up with this crap for the last 45 years. So, all right, that's the end. Yeah, go ahead.
What are your thoughts, right, as you mentioned, only homologous transcription factors for whichever species you're with.
Yeah.
What are your thoughts on how transferable between species?
Well, yeah, well, the thing with transcription factors, of course, is that how homologous are promoters is part of what you have to start to think about. And I've actually played a little bit with that, and it's surprising they are. There is homology, and it depends, but you can kind of imagine that, I was surprised when, baby boom, of course, it's in corn, it's in monocots, and so on and so forth. And there is some evidence where people have used transcription factors from one on another, but you're going to run into trouble there. That's why I thought the arabinogalactan protein was kind of weird, because we were using cotton on cacao and someone else was using it on tobacco. There's something there that's conserved that must be a receptor or something else like that. But there's no particular reason that the promoters have to be conserved, and that's going to be the governing factor for that, I think. So, oh, yeah, you try a bunch of them. Yeah, I mean, even us, even with what we were working with here in cacao, there was multiples. And so I showed you the one that worked, right? And didn't show you the one that didn't. And that's kind of how it goes. I mean, we didn't have any, we looked at them and they looked like the same. And not only that, if you look at AP2 domain transcription factors, they all kind of look a little similar too, right? And so we're like, is this really baby boom or not, right? And so that, I mean, it's a bit of a crapshoot. So that's why I think that's why I like the transient. Originally, when we developed oxytrophic strains for Monsanto, it was all about trying to do rapid plant biotech, right? Because you didn't want to wait years to know if a gene would actually function in that. And so that's why it's always been exciting to me is trying to better develop transient methods that are actually meaningful. And then you can do a test. And that's what we originally set up, because we wanted to use our propagation bioreactors, and we just wanted to flush transcription factors in, and then manipulate the morphogenic state, flush them back out again. And that was the intention of that design of the original NSF proposal, was the idea of doing programmed temporal development based on transient expression. Yeah. Yeah.
So when you're introducing UTR into the agrobacteria,
Yeah.
is the translation expression used more to ribosome stalling and slipping, or preventing recruitment from
that data is like a week old. Okay, so literally we were going back through, and when we found plasmids first, and we could do $15 for an entire thing, we just started to grab everything out of the freezer, and we're just sequencing it all. And then we started to look at it and go like, what's going on here? And we found a lot of things that we didn't realize were happening. And so I recommend if you go back and kind of look at what you really did when you cloned. And in the process of doing that, I'm under the impression now, having now read a bunch of papers on UTR regulation in bacteria, there's probably they're different transcripts. And so even defining, when someone defines a promoter, do they define it based on the transcription start site? Well, you don't know it, okay? And if it's different in bacteria than it is in the eukaryote, then what does it even mean? That's why on that slide, I showed the distance between the Tata box and the ATG, because it's kind of ambiguous what the actual transcript is in that case. And even in viruses, I was actually surprised that when you take a look at a Gemini virus, who's the virus guy? Okay, take a look at those and take a look at all the begomaviruses. When you align them, there's actually 2 to 3 in-frame start codons in the first 20, 30 amino acids. It's like, and so I asked the question of Gary Sunter and some of the guru. I'm like, well, which one of these is the right one? Like, I don't know. Probably all of them. And the reality is that probably gives some flexibility in the encapsulation of the virus to actually start the coat protein a little bit bigger, a little bit smaller. And that's probably how they evolve a little bit. And it can't be a coincidence. I aligned 10 different begomaviruses and like, look, there's this, they're in frame. Yeah, coincidentally, right? Yeah, right, they're in frame. Yeah, obviously it's not a coincidence. And when we did the proteomic studies, it turns out that we had purposely looked at the systemic activation of genes, but we did it very, very early because we were thinking we wanted to see what would happen before the virus got there. And then we went back and we're like, okay, so let's just answer this. You did all this fancy proteomics at PNNL, like where's the coat protein? And they said, we can't find it because we purposely chose the timeframes to be very early on in the process. And so we can't even go and use that data to figure out what the coat protein really is, because that would have been the obvious answer, would have been to just do it. And we could have, but we missed that boat. Yeah. Any other questions? Yeah.
I'm sure Dr. Rush might be using
Yeah, you and a lot of the world, right? I didn't, to me, I would just do it with nanoparticles. I would just launch the viral vectors with that and be done with it, right? I mean, DARPA, it turns out the DARPA director was an entomologist. Okay. You want the short answer, okay? And so the idea of being able to, now it was some really cool and interesting things that another reason I wanted to develop the actinic whiteflies is I wanted to come up with a system that you could actually do to make a conditional lethal. And when you have an insect that goes through 90% of its life as a nymph or they're attached to a plant, and so we were thinking we can't do hydroponics of the tet repressor, okay? And so the idea of creating essentially an actinic whitefly system was so that we now could potentially make a conditional lethal that would die. And so that was part of the thinking process here. But you're right. I mean, it is a bit scary and it flies away, and how do you control it? Well, if you had a really good conditional lethal, that would be one way of doing it. We were testing other things like having them track their way through entomopathogenic fungi. You see that connection to the game that's there, and like release them, but with a time bomb built in for something like that, with some of the ideas of dispersing the virus. Yeah.
Now I know. I think the note is online. So let's do it.
Sure.
Student sign for lunch.
Yeah. By the way, if there's anyone who wants to have, I'm going to bolt whenever it's right. But I don't have anything urgent, shall we say, except for, and I also have headlamps, so I'm perfectly comfortable hiking in at night. So if there is anyone that wants to sort of meet up and talk or that, you can certainly track me down as soon as I get my NSF fellowship application figured out. Yeah, so it's all fun until somebody gets hurt.
Just cover a lot.